Next, we're going to complete the biotechnology reading. Getting the idea. You might have watched a show about solving crimes using DNA evidence, or read about food being produced with ingredients that has been genetically modified. These are just two examples of the many areas to which the science of genetics has been applied. These include medicine, industry, agriculture, and crime solving. Selective breeding. Agriculture is the practice of growing crops and raising livestock for food and other products. Early in the history of agriculture, long before anyone knew about DNA or genes, humans began modifying organisms. They did this by selective breeding, the intentional mating of organisms to produce offspring with specific traits. Almost all domesticated animals and food plants are at least partly the result of selective breeding. For example, dairy breeders have selected for cows that give more milk. Breeders of apple trees have selected for size, taste, and color. In selective breeding, organisms with desired traits are mated. Their grown offspring who display the desired traits are also mated. This process is repeated over many generations until offspring consistently show desired traits. The process is slow. When people began to practice selective breeding, they did not know that the traits or genes were controlled by genes. Today, breeders can use their knowledge of genetics to produce organisms with desired traits more quickly. Selective breeding is actually how you've gotten all the different breeds of dogs and the different sizes, from the standard poodle that's a large dog down to the tiny toy poodle or the teacup poodles. They make the little teacup poodles. They take the smallest puppies from a litter and they breed those. Let them grow up. They breed the two smallest together. And then when they have offspring, they wait till they grow up and they breed the smallest of those together. And then they wait till those grow up and breed the smallest of those together until they get the smallest dogs that they can get. And of course, a lot of times those, after they've been bred so many times, they'll have health issues. But the selective breeding, that's that process of selective breeding to get those desired traits. Seedless watermelons are the same thing. They would take a watermelon with few seeds and breed them, and from that next generation, take ones with few seeds and breed them until they got seedless watermelons. Next, we have genetic engineering. Scientists have used their knowledge of genetics and DNA to develop organisms with traits that are useful to humans. This is one aspect of the field of biotechnology. Biotechnology is the use of organisms or biological systems to develop useful products and processes. Biotechnology has a wide range of economic and social implications. Genetic engineering is the direct manipulation of a cell's genetic material to produce organisms with useful traits. You're going to want to know that. Genetic engineering is used to produce organisms with useful traits. Genes or pieces of DNA from an organism of one species may be transferred to an organism of a different species. And organisms whose genes have been altered through genetic engineering are called genetically modified organisms, or GMOs. Selective breeding relies on natural processes. In contrast, GMOs cannot occur in nature. Genetic engineers have altered the genes of bacteria and yeast to produce medicines used to treat human diseases. They have modified lab animals to make it easier to study cancer and other diseases. They have improved some crops to make them more nourishing. They have modified other plants to be more resistant to damage from frost, insect pests, diseases, and herbicides. They've actually taken a gene from a fish that lives in icy ocean waters and put it into tomato plants to make them more resistant to frost, so you can grow tomatoes into the winter. Recombinant DNA. When the DNA of one organism is inserted into the DNA of another organism, the new DNA that results is called recombinant DNA. The laboratory process in which recombinant DNA is produced is called gene splicing. Recombinant DNA is often formed by transferring a portion of the DNA from a complex organism to a simpler organism. For example, scientists may insert a human gene into a bacterium. The following diagram shows the production of recombinant DNA in the common bacterium called E. coli. Refer to it as you read the next two paragraphs. Okay, this is an example of recombinant DNA. Maybe this is for human insulin, and this is a piece of a DNA from a human cell that contains the gene that causes the cell to produce insulin. Okay. This is the E. coli bacteria. Remember, prokaryotes and eukaryotes? Well, the E. coli is a bacteria, so it's a prokaryote. It does not have a nucleus. It does have a piece of DNA that's in a ring called a plasmid, and the scientists can remove that plasmid DNA, use an enzyme to splice or cut it open, take that piece of the DNA from the human cell that has that gene for the trait they want and insert it into that cut plasmid. And it reforms its ring. They can implant that ring back into an E. coli bacteria. And then it's going to reproduce its DNA material. That genetic material is going to make a copy of that. And then it's going to do binary fission, split in two. Then you've got two daughter cells that are identical. So they contain that human DNA, that human gene for making insulin. Now they're able to make insulin. And then they'll copy their plasmid, and they'll reproduce, and they'll copy their plasmid, and they'll reproduce, until you have a whole population of bacteria that are capable of making insulin. Recall that some of the DNA found in the bacteria cell is in the form of a ring called a plasmid. To form recombinant DNA, biotechnologists remove the plasmid from the bacterium, use enzymes to cut the plasmid, and then insert the human gene into the plasmid. Once the gene is inserted, the plasmid returns to its ring shape. The modified plasmid is then put back inside the bacterium. Recall that bacteria reproduce asexually through a type of cell division called binary fission. Binary fission is similar to cell division by mitosis, except that the DNA is not in a nucleus. Before reproducing, a bacterium makes a copy of its DNA, including the plasmid. If the plasmid contains recombinant DNA, the offspring will also contain recombinant DNA. This technique has been used to produce bacteria that produce human insulin. Many people with diabetes have to receive insulin because their body cannot produce insulin. Scientists have also used recombinant DNA to modify bacteria for industrial and environmental uses. For example, genetically altered bacteria are used to help clean up oil spills. The bacteria break down oil, producing water and carbon dioxide as byproducts. Other genetically engineered bacteria are used to produce minerals and make chemicals. Genetically engineered bacteria have many useful applications. However, the potential consequences of releasing genetically modified bacteria into the environment are an important consideration. Scientists must consider how genetically modified bacteria will affect and interact with the populations of living things in the ecosystem to which they are introduced. Recombinant DNA has also been used to change the traits of plants and animals. For example, goats have been modified to produce milk containing a certain protein. The protein can be extracted to treat people whose blood clots too easily. Some genetically modified sheep produce a protein that can treat emphysema, a respiratory disease. An example of a genetically modified plant is Bt corn. This corn has a gene from the bacterium Bacillus thuringiensis that produces a natural pesticide. Bt corn makes the same pesticide which protects the plants from the insect called the corn borer. Scientists have used a similar technique to modify cotton plants. The genetically altered plants are resistant to worms that attack cotton buds. This resistance increases the cotton yield and enables farmers to use fewer chemical pesticides. Gene therapy. Gene therapy is the alteration, insertion, or deletion of a gene in an individual's cells in order to treat a disease. Many genetic disorders involve missing or incorrect genes for making certain proteins. For example, children who are born with Tay-Sachs disease do not produce a specific enzyme necessary to digest lipids. This results in the buildup of lipids. This causes severe neurological problems. Scientists are looking for a way to use gene therapy to repair the DNA that codes for the enzyme. Gene therapy is still an experimental treatment. So far, it has had limited success, but many scientists think it holds promise for the future. Risk of genetic engineering. Like other forms of technology, genetic engineering has potential risk as well as benefits. For example, genetic engineering is used to create plants that create their own pesticides. If these plants are planted too widely or in the wrong place, they can harm beneficial insects. Other genetic engineered crops are resistant to weed killers. This is useful for farmers, but people worry that pollen from these crops would fertilize wild plants. This can result in larger populations of weeds that are harder to kill. A less obvious risk is that genetically engineered crops can reduce genetic variation. A genetically diverse population is often better able to resist disease and other threats. In the past, farmers in different places have grown many different regional varieties of corn, wheat, and other food crops. In some countries, a dozen or more varieties have been grown in the same small area. The scientists who are genetically engineering crops often create only a single variety with the desired trait. If all the farmers in an area plant the same variety of seed, they may grow more wheat or more nourishing rice. However, they may also increase the risk of losing the area's entire crop to a disease to which a different variety of wheat or rice would have been resistant. Cloning. Cloning is another form of biotechnology. Cloning is the artificial production of a DNA fragment, cell, or organism that is genetically identical to the original DNA fragment, cell, or organism. In some cases, scientists can use a single cell from an adult organism to grow an entire new individual that is genetically identical to the donor. Important thing to remember about cloning, the offspring are genetically identical. Cloning is not difficult when it involves making a DNA fragment because DNA normally replicates to form an exact copy of itself. It's also fairly simple to clone a single cell or a single-celled organism because each cell normally reproduces a daughter cell that's genetically identical to the parent cell. However, cloning in animals is a complex and difficult process. To clone an animal, scientists must remove and discard the nucleus from a female's gamete, the egg cell. Next, they inject the nucleus of a body cell from another adult into that egg. The two partial cells fuse and begin to divide. After a few divisions, the cell has become an embryo and is placed in the host mother. The embryo develops into an offspring that is genetically identical to the animal that donated the nucleus. This process is illustrated in the diagram below. Some researchers hope to use cloning to increase the populations of endangered species. Cloning may also be used together with recombinant DNA to produce medically or commercially valuable substances. For example, recombinant DNA could be used to produce goats whose milk contains valuable proteins. The goats could then be cloned to produce a new population of goats that would produce milk containing the proteins. Animal cloning raises some ethical concerns. A major concern is being able to clone animals could lead someone to trying to clone a human. Many people, including scientists, believe that cloning a human would be morally wrong. There's also the risk of producing an infant with severe birth defects. DNA fingerprinting. No two people's fingerprints are the same. Similarly, except for identical twins, no two people have the same DNA. Even identical twins may develop differences in their DNA during their lives. The same is true for other animals. This knowledge has led to a technology known as DNA fingerprinting. DNA fingerprinting compares images of DNA fragments to determine the relationship among organisms. Enzymes are used to cut the DNA molecule into smaller pieces, and the fragments are placed in a gel. There's an electric current is run through the gel. This technique is called gel electrophoresis. The fragments move through the gel at different rates based on their size. The resulting patterns of lines is called a DNA fingerprint. There's the DNA fingerprint. DNA fingerprinting technology has several uses. For example, DNA fingerprinting can be used to determine how closely related various organisms are to one another. The more similar the patterns of the two DNA fingerprints, the closer the relationship of the organisms. Biologists sometimes use this technique to identify related species and to identify and catalog endangered species. DNA can also be used to identify a child's mother or father. Forensics is the use of science and technology to investigate crimes and answer legal questions. Police departments and criminal lawyers use DNA fingerprinting as one form of forensic data. For example, technicians may collect samples of blood, skin, and other samples from crime scenes and make DNA fingerprints of the samples. Investigators can then compare the DNA fingerprints with the DNA fingerprints